Hello, everyone. My name is Dr. Rebecca Gilbert, and I am Chief Scientific Officer at American Parkinson Disease Association. Thank you so much for joining us today for our Dr. Gilbert Host series, where we are conversing with experts in Parkinson's disease to explore topics that are of interest to our Parkinson's disease community. Today's session has been generously supported by an unrestricted grant from Genentech. Today, we will be discussing Parkinson's disease and stem cell research. What is a stem cell? What can stem cells offer people with PD? My guest today, who will help us answer some of those questions, is Dr. Evan Snyder. Dr. Snyder is a trained pediatrician, neurologist, and neonatologist who spent 23 years at Harvard Medical School, where he established an independent research program and helped launch the fields of stem cell and regenerative medicine. In 2003, Dr. Snyder was recruited to Sanford Burnham Prebys Medical Discovery Institute and the University of California, San Diego, where he is now full professor, director of the Center for Stem Cells and Regenerative Medicine, and director of the Stem Cell Research Center. He is on faculty at UCSD School of Medicine, the Biomedical Sciences Graduate Program at UCSD, and Sanford Children's Health Research Center. He helped found the Sanford Consortium for Regenerative Medicine and is now on its scientific steering committee. He offers his expertise to countless organizations working in the fields of stem cells, and APDA is privileged to have Dr. Snyder on our scientific advisory board. So Dr. Snyder, thank you so much for joining us today for this topic.
Thanks for inviting me, Rebecca. This will be fun.
You're very welcome. I'm very much looking forward to our conversation. So first, we'll give an overview of what stem cells are so that everyone on our call or on our Zoom session is talking about the same thing. And we'll talk about why scientists are working with stem cells to help people with PD. After that, we will take questions. Now, some of the questions have already been submitted to us, but if you're watching this live and have a question, please feel free to enter it in our question and answer feature on your Zoom app. There are many people on our session today, so we may not be able to get to everyone's question. We will certainly try our best. So to begin with, let us give an overview of what stem cells are and why they are being investigated to help Parkinson's disease. So what are stem cells? A stem cell is a cell that is undifferentiated. What that means, it is a cell that's capable of developing into a number of different cell types. So the promise of stem cells is that these are cells that can be isolated, and we'll talk about how they can be isolated. They can be transformed in a laboratory into a dopamine-producing neuron, and then used to either study or possibly treat Parkinson's disease. There are many sources of stem cells, many more than two, but I'll discuss two now. The first are embryonic stem cells, and these are cells that are primarily isolated from embryos that are typically created for in vitro fertilization and not used. The embryos are not used, and the cells are isolated from the embryos. The second type of a stem cell that we will discuss today is something called induced pluripotent stem cells. So what are induced pluripotent stem cells? These are cells that are actually derived from fully formed adult cells. So as you can see in the left side of our schematic here, in the dish, in our schematic dish, are adult fibroblast cells. These are cells that can be isolated from a fully formed adult. These can be taken from a skin biopsy, for example, and the cells are taken into the lab and then reprogrammed with a series of steps that can be done within the laboratory. And they can be reprogrammed into cells that can then be differentiated into other cells. And so these, in this dish, are something called induced pluripotent stem cells. They're induced from an adult cell to become a pluripotent, meaning a cell that can become many other types of cells. And from there, in the laboratory, additional steps can be taken to create specific cell types, including dopaminergic neurons. So there's huge potential for using these cells, not only for treatment of all sorts of diseases, but, and maybe more importantly, to model disease. And we'll be discussing this concept in our discussion. Now, there are many, many hurdles to using stem cells of all types for transplantation into the brains of people with PD. And what are some of those hurdles? And we'll be addressing some of these in our discussion as well. What is the best type of cell to use? How do you ensure purity of the cells that are put back into someone's brain? How many cells to transplant? Where to transplant them? And how to make sure that the implanted cells don't go rogue and potentially form a tumor. And these are a lot of issues that are addressed or potentially addressed by scientists today. In recent years, there have been a few small trials around the world which have tested the safety and efficacy of various cell-based therapies for Parkinson's disease. Some of these studies await results, and we need a few more years to gather data to be able to present to the community about whether these treatments are beneficial, and more studies are in the planning stages. One such trial is a trial that's underway in Japan, in which dopamine precursor cells, which were derived from induced pluripotent stem cells, as I just discussed, are implanted into the brains of people with PD. A small trial is planned, and the results are going to be forthcoming. But what if there is a better way? What if we could create more dopamine neurons in a person's brain without actually inserting new cells into the brain? And that's a question that received a little bit of help in recent weeks, actually. There was a paper that was published in the journal Nature about 2 weeks ago. Another paper with a very similar finding was published a few months ago. And in these papers, the scientists showed there's a particular protein within cells that suppress the cell from becoming a neuron. And so these cells do not become neurons, they become other cell types. One such cell type is an astrocyte. This is a cell within the brain that is not a neuron. And so the researchers suppressed the suppressor and caused the astrocytes to turn into neurons. So this is a very exciting development. Of course, this work is very early. It was done in mice, but the concept that perhaps we could harness the potential of our own brains to create more neurons is very, very exciting. On that note, I'd like to open up our discussion to our Q&A and have Dr. Snyder answer some of the questions that we already received, and we await the questions that people will submit now. And so, Dr. Snyder, are you ready to answer our audience's questions?
I am.
Okay, fantastic.
Just take
So, okay, so the first question that we're going to try to address is, I have heard about stem cell clinics that are in the United States that are already treating people with PD with stem cells. Should I consider getting such a treatment?
Yes, I'm quite aware that there are a lot of kind of hucksters out there that believe that stem cell biology is easy, that stem cells are simply like sprinkling pixie dust on any disease and magically things will happen. And that just is not the case. Stem cell biology is part of fundamental developmental biology. And the approach that we in the stem cell field take is to try to recreate the system, but in this case, to do it correctly, or to compensate for what's missing. So in the case of Parkinson's disease, what you will see investigators doing is taking a stem cell, the earliest part of development. Just the stem cell is kind of a model of what happens shortly after fertilization, and then go through all the steps that nature does in making a dopaminergic neuron, but now to do it with a stem cell in a dish as opposed to what nature does in the body. This is a huge amount of intellectual heavy lifting. The clinics, however, have not done that, don't do that, and really have no idea whether they really have stem cells, where to put the stem cells, what the stem cells are doing. In fact, what they are doing is simply trying to extract money from you. So I come up with a little rule of thumb that if somebody, and this not only applies to the stem cell field for Parkinson's disease, I would say that this applies for the use of stem cells in any condition. If somebody is asking you to pay with your own money, it is by and large a sham. If it's a true trial, that will be covered. If it's a true treatment that's been approved, it will be covered by insurance. So that's a long way of saying that you should not go to those clinics. Not only will it not work, but there is the possibility of actually making things worse and creating a dangerous situation.
Okay, thank you very much for that clarity on that issue. I get asked that question a tremendous amount from patients. The next question is, so we're talking about how stem cells are still in the realm of research right now. Is there a way for our listeners to be part of a stem cell research trial?
Clinical trials, by definition, are experiments that are simply done in human beings. And experimental conditions have to be very, very regulated. Otherwise, the data and the findings that come out of it will not be interpretable. And of course, what you want at the end of the day is to have answers that say, this is the therapy that is safe and it is also effective, and these are the patients that qualify. So what you'll find is in most clinical trials, and they are very carefully regulated by the FDA, by internal review boards, by the experimenters themselves. Most clinical trials are very, very regulated as to who can be enrolled in the trial and who should be excluded and how many patients should be in each condition. And very often they're very randomized, so that even if one is in a clinical trial, you may be randomized to the control group, which means to receive nothing or to receive a placebo. So most of the time, you don't simply call up the investigator and say, can I be enrolled? Now, having said that, if you are being seen by a neurologist, you are within a catchment area of a particular academic center that is running clinical trials, and if your neurologist or the investigators know that you are out there, that you are willing to participate, they may contact you and say, you actually do qualify for this particular trial. Would you like to enroll? And then, of course, you sign consents, some of which are that you may not be the one receiving the treatment in question, or we don't know whether this completely works. It may not work. It may not be entirely safe. Are you willing to accept that risk? So the short answer is you don't necessarily have the ability to hear about a clinical trial halfway across the country and say, I want that. On the other hand, if you are part of a clinic, a movement disorder clinic, a Parkinson's clinic, it is possible that you may be tapped to participate if you qualify for the inclusion criteria and are not part of the exclusion criteria, which can also be very rigid.
Excellent. So anyone interested in stem cell research should talk with their neurologist and see what's available. And I think that's fantastic. And we have a question here that actually reminds me of something that you and I discussed in preparation for this, which I think would be an interesting question to ask you now, which is, this is from Leo. PD is not only a disease of dopamine deficiency, but of other neurotransmitters. So will dopamine programmed stem cells alone be helpful?
Oh, that is
excellent question.
That is an excellent question. And it gets to the heart of so many issues in the Parkinson's field, including the stem cell field. I'm not even sure whether I can give you a short answer, but I will try. You are absolutely correct that we in the field of Parkinson's disease are starting to recognize, it's always been there, but now we're confronting this, that Parkinson's disease is not solely a movement disorder. It's not solely a disorder of dopamine neurons. It just so happens that dopamine neurons kind of are the target, or the most prominent first target of whatever this process is that causes it. And we still don't know the initiating event. There's all kinds of theories about what the initiating event is. Is it bad genes? Is it a bad early environment? Is it the wrong microbiome when you were born in your gut? Is it an inflammatory reaction? Is it a viral reaction? And to an extent, what we would love to do is go down to the big bang of Parkinson's disease. And that is actually one of the areas that the stem cell field is investigating. For example, Dr. Gilbert mentioned that these IPS lines are also being used to model Parkinson's disease in a dish, to look at a neuron or the other cells in the brain and say, I took this from a Parkinson's patient, therefore I have neurons in a dish that have Parkinson's disease. Let me study it, let me figure out how it started, let me find a drug target, and then discover a drug against it to stop the disease from ever happening to begin with. Maybe even start treating patients at risk before symptoms become apparent because they're at high risk, or it runs in their family, or they have certain markers for it. That is still very, very, very much in the future, though that's an area that we are actively investigating. On the other hand, the history of Parkinson's disease treatment has been addressing what are the most prominent symptoms, which is a dopamine deficiency, which leads to a movement disorder. And this discovery, surprisingly, is not as old as people would imagine. The notion that even though the movement disorder has been known for quite a while, since James Parkinson, that it's due to dopamine only dates back a little over 50 or 60 years. And the notion that replacing dopamine can ameliorate these movement disorder symptoms is also only about 40 or 50 years of age. And all of this is still very new. The notion of emulating with cells what is being done with drugs only dates back to the 80s. And then finding better ways to generate these cells, not just clumps of cells from a developing brain, but also making them in a dish, that dates back only maybe about 15 years or 20 years and is still actively going. We do recognize, however, that when we replace dopamine, we are probably addressing just the tip of the iceberg. We are not stopping what caused the disease. We are not possibly even stopping progression of the disease. But we are attacking what most patients will say is the most vexing symptom that they experience, this inability to maintain the same activities that they've enjoyed throughout their lives. So that's a major win, but we do recognize that Parkinson's therapy focusing solely on the dopamine system may not address some of the cognitive problems that somebody might be experiencing, or the emotional problems, or even some of the gut problems and other kinds of endocrine problems. So it's a beginning, it's an important beginning, and it's an important target, but the questioner is absolutely right that we need to think bigger, and we need to start coming up with therapies that address all of the non-dopaminergic manifestations of this disease. Now, we're very confident that the stem cell field will actually help in that regard. One, we can make stem cells into other kinds of neural stem cell type, and other kinds of neuronal types that make other kinds of things. We can arm them with various genes and proteins that can protect other kinds of cells, make other kinds of neurotransmitters. And then, as I said, we can use the stem cells to simply model the disease in a dish and see all the other manifestations and then use them for novel drug discovery. But that not only is an excellent question, that question defined what is probably the next 25 years of Parkinson's disease research.
Absolutely, and I think it also highlights, as you were saying, how complex Parkinson's is, and even though it seems that stem cells is a really complex solution for a really complex problem, it's not complex enough to really deal with the complexity of Parkinson's. And it actually helped me answer one of the other questions that came in, which is, and I don't know much about this, but the questioner is saying that she has seen stem cells used for orthopedic indications and successfully, and is hopeful that it could be used for Parkinson's itself. And I think our previous questioner highlights that obviously neurologic diseases are much more complicated than orthopedic diseases. I mean, as you know, difficult. I don't want to insult any orthopedists out there, but that is the bottom line. So perhaps you can elaborate to help that questioner. A questioner who's referring to the orthopedic uses of stem cells. What are the hurdles specifically for neurologic diseases that may not come into play when we're modeling or using stem cells to treat diseases that are not neurologic?
Well, again, this is kind of a two-part question. And to tell you the truth, this confronts physicians dealing with any disease. What you are confronted with is a patient who has symptoms and signs and complaints. So of course, your first job is to relieve suffering, and that usually means relieving the symptoms. And in a way, that's what stem cell biology has been doing for the last 30 years, addressing the symptom of a movement disorder, which looks like it can be tied to a dopamine deficiency. In the same way in orthopedics, what the questioner is alluding to is the fact that if you have an orthopedic problem, you have arthritis in your knee, in your shoulder, in your hip, what it appears is that the stem cells that are being administered in those situations is designed to relieve the inflammation. However, somebody could say, well, that's great, and I am fine with getting a new stem cell treatment to relieve inflammation every 6 months or every 12 months, the same way I'm used to getting a steroid shot or something like that. It doesn't answer, why did you get arthritis to begin with? Why in the world are you having an inflammatory reaction? So the same thing can be with Parkinson's disease or any neurologic disease. It could be, and we're trying to figure out which patients with Parkinson's disease would benefit most from dopamine replacement, whether it's with a drug or a cell or a gene. And we think we know. It's not all Parkinson's patients. It's those who do seem to respond to receiving dopamine back. Again, whether it's with a drug or some other way. But the other, and a patient could say, you know, I am totally happy with that. If you just allow me to address this symptom, even if I need to have, you know, recurrent treatment, I am happy with that. And that probably is until that stops working or the other manifestations of Parkinson's disease start becoming prominent, which gets to the other thing that we in the field want to do, is to stop the disease from progressing, maybe even stop the disease from finding expression to begin with. And that gets down to trying to understand, as I mentioned before, what triggers the disease? What starts it to begin with? And can we identify those at risk and nip it in the bud, stop it from ever developing? And there's a whole area of research that has nothing to do necessarily with dopamine neuronal development. It's looking at the various kinds of neuronal cells that have kind of the fingerprint of what we know seems to be associated with Parkinson's disease. And some of the listeners may be familiar with Lewy bodies. This is an accumulation in many cell types of a protein that just doesn't get broken down appropriately. So some investigations are trying to figure out, is that the cause of Parkinson's disease or just the signs of the disease having started? Others are investigating whether maybe this is a fundamental defect in the trash disposal mechanisms of the neuron. There are various, what we call organelles, or kind of structures inside a cell that are meant to clear toxic proteins. One of those may be familiar, they're called lysosomes, and they package and degrade protein. It may be that that factory that is meant for waste disposal is just not doing its job correctly, and there may be genes that focus into that. And then it looks like when neurons and cells talk to each other, something bad happening in one cell may transmit that to another cell. So a whole area of research is not dealing necessarily with the symptoms of Parkinson's disease. That's definitely important. That's what is vexing patients, but is also investigating what started it to begin with. So we are looking at cells that may not even be dopaminergic neurons. They may be neurons in other parts of the brain. They may not even be neurons. They may be the support cells called the astrocytes. They may even be the bacteria in the gut or gut cells, to see what made the neuron's trash disposal system not work to begin with, and can we find a target and then a drug against that target. So again, that's a long answer of saying that our world, our stem cell world, but also the Parkinson's world, is divided up into two different camps that talk to each other all the time. One are the symptoms, the other is addressing the cause. And hopefully the symptoms, I think, we are starting to make some progress on. I think, as Rebecca mentioned, there are clinical trials that look as if they may be able to address the symptoms that are due to dopamine deficiency. At the same time, in parallel, not quite as advanced, are efforts to figure out what started the disease, and are there ways to detect who's at risk and start treatment early on and prevent it.
Fantastic. That's very, very clear. I'm asked a question here, Gerald asked this question, that he has heard, or he thinks that stem cell replacement will be effective for a short period of time, but may not be effective long term. Do we have any information on that question?
Well, that also is an area of debate, and that falls down into a few sub questions to that. The first is, how long, even if you make neurons that make dopamine, how long will they make dopamine? How long will they last in the brain? And will they themselves get Parkinson's disease? The evidence that we have so far, and this is based on some of the studies that were actually done back in the 80s using dopamine, early dopamine precursors from fetuses that were implanted in a patient's brain, is that they do not seem to be rejected by the immune system. They do seem to last a very, very long time and continue making dopamine. They do seem to make connections, and these cells seem to confer benefit for a very long time. And by a very long time, I mean up to 15 or 20 years. That's the age of some of the patients that receive those early transplants. Now, will dopamine replacement ever stop working? Now, that is a possibility because we know two things. One, that patients with very severe progressive Parkinson's disease ultimately do find that their medication doesn't work as well. And we know that the cells do basically what the drugs do. It also does not stop the progression of the disease. As I mentioned, one of the investigations that the stem cell field is also doing is what triggers the disease and what makes it progress. And we do know that stem cell transplantation, making dopamine, does not stop that progression. So there is a period of time where it may stop working. Now, another part of the question, as alluded to, is can the cells themselves get Parkinson's disease if they've been around a long time? And could that be a reason for them to stop working? And this is also is a hot debate in the field. It does look like some of those cells that were transplanted back in the 80s, when now some of the more elderly patients are starting to die and one looks at the brains, it does look as if not all groups, but some groups found that these cells did have the signs of Parkinson's disease. They had these Lewy bodies. On the other hand, it was not all of them, and not all of these neurons, and it was somewhat invisible to the patients. Even if these cells got signs of this disease, it didn't impair the improvement that the patients experienced. They continued to derive benefit from these cells. And then, of course, one could say, well, maybe after 20 or 30 years, maybe these cells really will start suffering from Parkinson's disease, the way a patient starts suffering from Parkinson's disease, maybe in their 40s or 50s or 60s. And somewhat the answer to that is, well, even if you need to get new cells after 20 or 30 years that are also good for another 20 or 30 years, maybe that's not a bad investment. Maybe that really is just like, at the end of the day, changing your transmission on your car, every decade or two. So it is a hot debate. It's a great question. We do think that at some point, for some patients, there may be an end to the efficacy of these cells. We don't think that there's any danger, but hopefully by the time that happens, the other side of my field, looking for drugs that can stop progression and that can stop onset, will have caught up and we'll be able to do combined therapy.
Excellent. I have another question, which I think is ripe for discussion, which is, how do transplanted cells make neural circuits once they're implanted in the brain?
Oh, that really is also a really excellent question.
I told you that we would get amazing questions, and we have been.
And the reason I love that question is that it really shows a level of sophistication that I would say most physicians and scientists and even stem cell biologists don't entirely have. And the heart of the question is, it's not really enough to say, I have a nerve cell, I'm going to stick it in there, and it's going to live happily. The questioner obviously recognizes that the brain is really like a telephone network. The way the brain works are not neurons in isolation, but making connections with other nerve cells so that information gets transmitted and signals get transmitted, and that even the support cells don't exist there in isolation. They're constantly communicating with the nerve cells. They're detoxifying them, they're providing nutrition and growth factors and supporting the environment. and blood vessels course through there to provide nutrition and to remove also toxins and things of that sort. It is an entire village. It does take a village. Now, we in our field, embarrassingly late, have realized it's not enough to turn a stem cell into a neuron, stick into the brain, and then go, voila, aren't things great? Here's my neuron. What you want to do is see connections. And it looks as if, and this field is still very, very new, that the fetal tissue cells that were placed, again, back in the 80s, when they've been looked at, in some cases, 15, 20, 25 years later, were making connections. And there's some evidence, not as old, that neurons derived from stem cells, when they're implanted and you wait a long time, are making connections. And we can study how these connections are formed and how they function. First, we can simply trace them by putting in dyes. But then we can also do various kinds of tracers that highlight whether these cells are really making true functional connections called synapses by using certain kinds of dyes that will recognize these connections and actually jump across to show that there is a connection. And then there are ways of showing that these connections are functional by making sure the neuron actually has a switch that can be turned on and turned off, either with light or with a drug, to show that the neuron is there, it's making a connection, the connection's functional, and that function is necessary for the improvement, for example, that an animal model is seeing. You turn off that connection and the animal's Parkinson's disease symptoms come back. You turn on the neuron and the function seems to improve. So the question is right at the heart of what stem cell biologists are now focusing on. It's not enough to make a neuron. It's maybe not even enough for it to spew out dopamine, though, you know, we'll go with that. You really want the connections to be reconstituted that went awry to begin with in the disease. Excellent question.
Very good. So you have another question that just came in, which is, are fetal cells being used? And perhaps we can use this question to highlight what the difference is between fetal cells and embryonic cells. What is the status of those two different kinds of cells?
That's also a really good question. It also dips right into the heart of the debate that stem cell biologists and cell therapists are having in the Parkinson's field. So I think most of us would say that nature is the gold standard. Everything we do in the stem cell field is simply trying to emulate what nature always does best. So the early areas of the cell-based therapy field for Parkinson's disease was going to fetal cadavers. In other words, for whatever reason, a fetus that somehow didn't make it to term, that died early in the womb or early out of the womb. What investigators did was go to the part of the brain that makes dopaminergic neurons. Again, knowing nature still does it best. Take those cells, put them into Parkinson's patients. And those did seem to work. Now, there were complications with that because it's not a pure population. There were contaminations because, again, you're just going with whatever you have. And we can talk about what those complications were later and what kind of held up the field for a very long time. But I think we always realized that there were always going to be more Parkinson's patients than there ever was going to be availability of fetal tissue. Fetal tissue comes unpredictably, serendipitously, and usually with not providing enough cells for all the patients that need it, maybe not even for a single patient that needs it. You probably need, for a Parkinson's patient, you may need as many as 3 to 6 exact fetuses and their tissue per patient. And as I mentioned, it's not a well-characterized pure population. There were some side effects in some patients, and we thought it was due to some contamination. So while it was the gold standard, from the very beginning, we sought ways to try to not have to use fetal tissue that would provide pure, well-characterized, abundant amounts of dopaminergic neurons. And that's what the stem cell field allowed us. But again, to make these dopaminergic neurons, we simply tried to understand what nature's playbook was, what the recipe was, and simply following it. And we're still trying to improve on the recipe, but it looks as if the stem cell field will try to be what the distillate of fetal tissue is. Now, what was a little bit of a caution to me by one of my colleagues was that we in the stem cell field have still never quite achieved the excellent recovery that the old fetal tissue field was able to achieve. So when I was a young Turk, I used to think, well, we young stem cell biologists are going to put the fetal tissue guys out of business because we're the next generation, we're the next wave. I've now become humbled in realizing what we need to do is learn from the fetal tissue field. Now the question is there still fetal tissue research going on, actually taking the cells and putting them into patients? And the answer is yes, for exactly the reason I mentioned, that as we in the stem cell field are trying to get better and better and better and come closer and closer to emulating nature, and we'll do clinical trials, some, as Rebecca said, are already in process, there are some who do believe that we still have not abstracted all of the knowledge and benefit that the fetal tissue field found back in the 80s, and that maybe it's time to do those studies again, but better, with up-to-date techniques and up-to-date surveillance and up-to-date preparation and administration and homogenization and regimentation. So there is a trial ongoing in Europe where they are redoing, but much better and much more carefully, the fetal tissue trials now that were done in the 80s. They've done so far 11 patients, and starting about 3 years ago, and they are carefully following these patients. So it's interesting that in a way it's back to the future. 2 areas of research are going on, again, in parallel simultaneously, and hopefully with a lot of crosstalk, everybody sharing their data to figure out how to do things better and what to learn from each other.
The questions keep coming in, and they're all fantastic. It's going to be impossible to answer them all. So I just want to mention anyone who has submitted a question that we do not get to. I apologize in advance. I will try to answer your questions offline. So stay tuned for an email from me in the next few days. But there are a couple that I do want to get to for the benefit of everyone. This next one is, what about umbilical cords as a source of stem cells? What are your thoughts on that for PD? That would seem like there is an unlimited supply. People have babies every day.
It is true that umbilical cord, blood and the umbilical cord itself does have kinds of stem cells. Mostly these are blood stem cells. And to my way of thinking, stem cells coming from the umbilical cord, blood, are excellent for therapies that can be treated with the equivalent of a bone marrow transplantation, or that are blood-based, or that can have gene therapy based on that. They can even be turned into these IPS lines that Rebecca talked about before. But to my way of thinking, umbilical cord stem cells straight out of the cord have no place in Parkinson's disease therapy. They will not work.
Thank you for that answer. One questioner mentioned the UCSD paper that I brought up in the introduction and was asking about your thoughts on applicability to the clinic. So what are your thoughts on the paper that I mentioned? Maybe recap a little bit if people didn't catch the intro or missed what that was about.
Yes, well, again, that's an excellent question. In fact, it's the question that my former grad student texted me when the paper came out. He literally said, "Did you see that paper from our old institution, from UCSD? Do you think that's a game changer?" So the questioner asked the question that my own former grad student posed to me a week ago. And the answer is, we don't know, but it is intriguing to think about it. Now, for those who weren't familiar with what the paper did, as I mentioned, or as you may know, in the brain, there are nerve cells, and those are the business end of the brain. Those are the ones that fire the electricity, that make the connections, that produce the dopamine. And then there are other cells that support these cells. These are called glial cells or astrocytes or support cells, and they kind of nestle the neurons. Now, what we do know in the stem cell field is that they all come from one granddaddy cell. And then as development happens, they split, and one becomes the worker bee and the other becomes the helper cell. What this paper did was to say, well, since they all came from a common ancestor, maybe we can simply make the helper cell become a worker cell and just suppress that signal, whatever the signal was, and they think they know, that caused this division of labor, just suppress it and make the helper cell turn into a worker cell. So now, if the workers are getting diseased or depleted or disappearing or dysfunctional, just take a fresh support cell, let them fill up the ranks, let that become a worker cell. In other words, turn an astrocyte into a dopaminergic neuron. Wouldn't that work? And all you need to do is give a single drug that blocks that switch. So you go back into becoming a worker cell. It sounds very appealing. It sounds as if it could work, but like in most things in science, it's not always the first paper that's most important in the field, even though that's what gets all the headlines. It's the second paper in the field. Somebody else duplicating the results and then showing that it can work in other kinds of models of Parkinson's disease that maybe emulate more what actual patients are experiencing. Larger animals, different kinds of symptoms in Parkinson's disease. So the short answer is intriguing, a novel approach, very enticing, could come down to a single drug, though of course we need to see, what we tend to do is look what's going on under the lamppost. We don't look what's going out there. If we give that as a drug, are there other bad things that are going on? We don't want that kind of conversion, but that's what science is all about. Replication and looking for all of the good and not so good effects of it and showing that it's reliably reproduced by many different labs in many different animal models. And then it could be that in a few years, we will see a clinical trial of that sort.
Fantastic. Just to address a question that has come up a number of times, which is, what would be the time frame that people should expect at this point? If a treatment is developed where stem cells are used, injected into people's brains with Parkinson's to help their symptoms, what would the time frame be to be a treatment that's not a research protocol anymore? Your best guess.
So I guess the question is not even a clinical trial, but
Right.
an FDA approved
FDA-approved, yeah.
that any neurologist can refer a patient to receive. I would say this kind of intervention, a transplantation intervention, becoming standard of care, where all of the clinical trials have been finished, the patients have been followed up to make sure that there are no lingering long-term undesirable side effects like seizures and excessive movements and tumors and things of that sort. I would say that that is still probably about 10 years off from becoming standard of treatment that any neurologist can recommend for their patients.
Okay. I have another question that actually came in before this session, which I want to address because it was a really good question. And if treatment does proceed along these lines or during clinical trials, would stem cells used to treat Parkinson's disease need to be from a matching donor, or is it better from the patient himself? If the approach is the iPS cells, is it better to use cells from the patient themselves or from a matching donor? And if they're used from a donor, would a person theoretically need to take an anti-rejection medication once they've received their transplant?
Well, the listeners are asking great questions.
Great questions.
They're great questions because these are the questions that make for absolute Talmudic debate in all the meetings I go to, because there is no set answer yet. There's just a lot of data coming from different groups that needs to be weighed. So if one goes back to the early days, as I mentioned before, of the fetal transplantation world back in the 80s, what those investigators did was do various kinds of immunosuppression that ranged all the way from zero immunosuppression to emulating what was done with organ transplantation at the time, hearts, kidneys, things of that sort. And it looked as if, based on that, that minimal immunosuppression was needed. That none, in some cases, none was used and the cells survived. In most cases, it was not long-term immunosuppression, maybe just for a few months. So the thinking was that no immunosuppression was necessary. And that at the time fit in very nicely with our thinking that the brain was what we say immunoprivileged. Unlike other parts of the body, it was protected from immune attack. We now know that that's not necessarily the case, that there can be immune attack in the brain. We simply don't know to what extent that can be. So we are now in the area of investigating whether we do need to worry about immunosuppression. So there are various regimes being studied. There is the assumption that we are not going to be able to escape scot-free, that we're going to have to do something. So now we're dealing with the balance of recognizing that maybe it is best to take cells from the patient, him or herself. That's completely matched. That's called autologous. And of course, the iPS field allows that to happen. You can pull, as Rebecca mentioned, you can pull a skin cell or a blood cell and turn it into an early stem cell and then turn that into a dopaminergic neuron. But that is a lot of work. There's a lot of variability. And there's no guarantee that if you do that for a patient in California, that the patient in Massachusetts will be getting exactly the same quality cell. So this kind of gives drug companies and scientists and cell cell companies heartburn to think that there can be this variability and not good quality control. What everybody wants is something, an off-the-shelf, pure, well-characterized, safe, effective, homogeneous product. So that you can take a cell, if that turns out to be the therapy, the way you pull off a bottle of Tylenol from the drugstore, and you know that it's going to be okay. So then how, if you're not going to be able to literally make dopaminergic neurons from each patient, now maybe we will get good at that. Maybe that process will become so routine that you do get that product. We're not there yet. So now we're dealing with, all right, we can't have an individual cell bank for every single Parkinson's disease patient that's there. That's not practical, cost effective, or maybe even safe and effective. How do we deal with stem cells that are good for all patients? And we call that allografts as opposed to autografts. And now we try to figure out what is the risk of there being immunorejection. So every kind of strategy is being entertained. Maybe it's just certain molecules on the surface of the cell called HLA antigens or human leukocyte antigens. That's what triggers it. Maybe we just need to cloak some of those. Or maybe put in universal donor sites on the stem cell. Or maybe what we need to do is short circuit any kind of immune reaction in the cells themselves. Or maybe we need to do co-grafts, use the cell, but encase it in other cells that make various kinds of immunosuppressant molecules all the time. Or maybe you do just do short-term drug therapy, or maybe you don't need to do anything. So this area is, and it's called neuroimmunology, is actively being investigated by everybody in my field thinking about cell-based therapies for the brain, not just for Parkinson's disease, but for any disease. But it looks as if, I would say, for any transplantation target in the stem cell field, Parkinson's is way ahead of the game and probably is turning into the poster child for every other field in neural transplantation.
Well, that's good news for our audience. So thank you so much for that answer. It is 5 to 1, so we're going to wrap up now. Again, we have a number of questions unanswered. That's the nature of this topic. So to summarize, we have an extremely complex potential treatment that's undergoing rigorous research right now throughout the world. And but not only are stem cells being investigated for transplantation purposes, but they are a treasure trove of potential modeling of Parkinson's disease to figure out, as you have mentioned many times, that it's not just the treatments of Parkinson's with returning dopamine, but figuring out the core source of what Parkinson's disease is and why it happens, and potentially stem cells can help with both in their own way. So thank you so much for all your insights. And we want to tell everybody who's listening today, if you missed part of this session or you want to share this session with someone else, a recording will be available on apdaparkinson.org and then over the next few days, and will be on our YouTube channel. So please feel free to share that or to listen to it again. APDA has many valuable resources. And here is a selection of some of the resources that are pertaining to stem cells and stem cell research. And if you would like to access this, we will be sending this list of resources to the people who participated in our session today. Now, please consider donating to APDA by visiting our website at apdaparkinson.org so we can continue to bring you programming like this to help you understand your Parkinson's disease and to help you understand potential treatments and research that is out there. So on that note, again, I'd like to thank so much Dr. Snyder. Again, Dr. Snyder is on our scientific advisory board, helps us select our grants, really helps us as an organization. And thank you so much for giving of your time and expertise today to highlight these exceptionally important issues that clearly our Parkinson community is eager to hear about. So again, thank you so much. Our next Dr. Gilbert host session will be on August 10th, again at noon Eastern Standard Time. And at that point, I'll be conversing with Bill Rasmussen. He is the founder of ESPN and a person with Parkinson's disease. So it'll be an entirely different kind of conversation, but it should be very interesting as well. So stay tuned for that. Thanks again, everyone who is on our call and on our Zoom session for joining. And we hope to see everybody again very, very soon. Thank you.